Introducing Gemma Barossi. Oh, hi. What an introduction. So dark down here. Anyways, my name is Gemma Barossi. You can call me Gemma for short, but please don't confuse me with my direct relatives, G. indica or G. pacifica. I don't know much about them. We never interact. Actually, I don't really interact with any other living organism now that I think about it. Never call me strain 121. I feel like I sound like a disease, and I actually never cause any to humans. As a matter of fact, my first encounter with a human was in 2003. I was found in this deep sea hydrothermal vent along the Endeavour segment of the Juan de Fuca Ridge in the Northeast Pacific Ocean, 200 miles off Puget Sound in Washington State. The reason for all the black smoke in this vent is because magma heats up seawater that gets into little cracks along tectonic plates. This chemical reaction causes mineral deposits. So this black smoke is actually iron sulfide. As you can see, I'm coccoid in shape and I have several lophotrichus-like flagella. Like most Archaea, I keep it simple with a cytoplasmic membrane, periplastic space, and a single outer surface layer. My lineage goes back a long way. We'll go over the boring details first. Let's see. My domain is Archaea. My phylum is Archaeota. Class is Thermoprotea. Order is Desulphurococcolese. Family is Pyrodictiae. Finally, genus is Geogemma, and my species is Barossi. A fun theory I've heard since my discovery is that scientists were able to compare the sequences of the 16 sRNA among prokaryotes who exhibit the most primitive characteristics. They confirmed they are all hyperthermophiles like me, which made them hypothesize that perhaps life first occurred in deep sea hydrothermal conditions, especially considering way back then volcanic and hydrothermal activity was more common. I believe this. I won't let it get to my head though. I'm so smart. So yeah, it sounds like I'm kind of a big deal. Apparently, I hold the record for growth at the highest temperature for any living organism. Thank you, thank you. I grow the fastest at 103 Celsius, but can handle 121 Celsius. If the temp reaches 130, I'm still alive. I just stop reproducing until it cools down a bit. But I heard that someone is trying to steal that title, Methanopyrus candelier, just because they can grow at 122 degrees Celsius. I don't even think they deserve that title at all. Their optimal growth temp is 98 Celsius, which is so much lower than mine. Okay, back to talking about me. It's difficult to replicate my metabolism in a lab since I am an obligate chemolithoautotroph who cannot use oxygen. CO2 is my carbon source, and iron is an inorganic compound that I use as my final electron acceptor. I reduce ferric iron to ferrous iron, and magnetite is a byproduct, which is obviously why I live near these vents. The process of reducing iron oxide through respiration is actually a really important role in the biogeochemical cycling of iron. It influences the solubility and mobility of iron in various ecosystems. You see, iron exists in mainly two different states. Ferric iron in an oxidized state is difficult to dissolve at ambient pH, but ferrous iron in the reduced state is easily soluble. I'm not the only one who enjoys iron. Its reduced ferrous state is actually a vital micronutrient for phytoplankton. Phytoplankton also absorb a lot of carbon dioxide through photosynthesis, essentially regulating the ocean's carbon cycle and reducing atmospheric carbon.
Accessing files.
Oh no! It's happening! Everyone, stay calm! No need to be alarmed. It's just binary vision.
60%.
So, I've concluded that not only do I manage to maintain the marine iron cycle, but also influence the carbon cycle on the entire planet. Oh, and my great ancestors might have been the first living organisms on the planet. Wow, thanks for coming down. Bye.
Hello, my name is Gareth Fielking, and this is my term project presentation on methanogenic archaea and the profound effect they have on our world now and in the future. Microorganisms help shape almost every aspect of the world around us, from disease processes to processing the decay of fallen leaves, manipulation of microbes for food and production, to medical innovations. They have a profound effect and role in propping up modern society. However, no group of microorganisms is poised to have such a large effect on the globe and human population as methanogens. Methanogenic archaea that exist in the rumen of beef cattle and ovine cattle generate a high amount of methane that affects the atmospheric composition. In the future, methanogens that have been locked up in permafrost and marshlands may release enough methane to cause a tipping point event in the balance of our global climate. Methanogens are a family made up of almost exclusively archaea that are found in soil, lake beds, and deep oceanic vents. Most methanogenic archaea are from the family Methanobacteriaceae. This family includes Methanobacterium, Methanosarcina, Methanococcus, and Methanothermobacter. This last entry includes a variety of archaea that exclusively exist in deep sea hydrothermal vents and may have been some of the earliest prokaryotes to evolve on Earth. Methanogenic archaea can exist in almost as wide a variety of shapes and configurations as their bacterial counterparts, including Strepto and Staphylococcus. They can also exist in Streptobacillus configurations, which is the primary way they exist in the rumen of cattle. Methanogenic archaea's major difference is that they lack a peptidoglycan cell wall. Other substances exist in peptidoglycan's place. Most methanobacterial genera utilize a partial reductive citric acid cycle. During their cellular respiration, they primarily generate methane as a product of the fermentation of acetate, specifically activated acetate in the form of acetyl-CoA. This pathway is largely used in archaea that are strict anaerobes. These bacteria can synthesize a compound called carbon monoxide dehydrogenase, or CODH, which catalyzes the cleavage of acetyl-CoA. The resulting methyl group is eventually reduced, partially producing methane and carbon dioxide as byproducts. This metabolic loop is enhanced when the methanogenic archaea are in a system with other bacteria that are acetogenic, such as Clostridium. In this way, there is much more acetate around to ferment, which is the exact environment that is present in ruminant animals. Livestock and manure from that livestock account for 5.8% of global carbon equivalent output from human sources. I include this as a human source because it is due to the high global demand for beef and ovine products that necessitates the existence of such a large population of large ruminants. This output is driven by the methanogenic archaea not just in the rumen of the ruminant animals, but also in those that live in the decomposing waste ponds that border large cattle operations. Biowaste consumption, after all, and its disposal is one of the industrial and agricultural applications of methanogenic archaea. In fact, the top four agricultural sources of methane come from, in this order, beef, lamb and mutton, cheese production, and dairy herds. The majority of methane in this process is produced during the farming phase of production, meaning it is solely a byproduct of the methanogens at work in the rumen of the cattle. Fortunately, there are some mitigation strategies that can be employed to help with the production of methane from ruminant cattle. These methods include vaccination of cattle to limit methanogenic archaea, or introducing more dietary fatty acids to their diet, both of which target the archaea directly to reduce their methane output. Of course, the best way to reduce the impact of methane from this animal food system is to reduce our consumption of meat. It isn't a large percentage of the total carbon equivalent output, but it is significant and it is low-hanging fruit in the fight against global climate change. Thank you for listening, and I hope you found this presentation at least marginally informative.
Imagine all the world's greenery returned to violet, to a time when our ancestors had not yet dreamed of chlorophyll, and when all the sun's light was harnessed by retinol instead. Science fiction or fact? The answer lies in the extremes of life. For my term project, I have chosen Halobacterium, a genus from domain Archaea whose closest relative is the methanogens. Halobacterium is of particular interest to astrobiologists who are searching to answer one of life's biggest questions: are we alone in the universe? Astrobiologists are particularly fond of Halobacterium because species members are polyextremophiles. This means that they are adapted to survive in multiple extremes under multiple stressors, like those that may be encountered in planets outside our own, like the surface of Mars, for example. One of the most fascinating features of Halobacterium is that they produce spectacular blooms that are visible from outer space. And some scientists believe that when oxygen levels on Earth were low about 4 billion years ago, Halobacterium may have painted the Earth's surface purple long before chlorophyll painted it green, and you may have noticed it from outer space. As the prefix in the name implies, Halobacterium thrive in hypersaline environments. However, the suffix bacterium in this case is a misnomer, since scientists now know that this microorganism is not a bacteria but a member of Archaea instead. They share many common features with other members of Archaea. They are pigmented, they are bordered by a lipid monolayer containing ether-linked phospholipids enclosed within a glycoprotein cell wall. They are flagellated, which allows them to swim to places where growth conditions are optimal. They are generally rod-shaped, but some species members are highly pleomorphic and can exhibit some unusual shapes. And rather unique to them is the presence of glass vesicles that allow them to float. Salt is of course required for growth. They are easy to culture in a lab setting, as they can be grown from liquid culture, colony to liquid culture, and even cell colonies. They are metabolically flexible. That is, they can grow both in aerobic and anaerobic conditions. Under aerobic conditions, they can obtain energy through cellular respiration by oxidizing various metabolites. And when oxygen levels are low, they can generate energy through fermentation of the amino acid arginine. But their most unique characteristic is that they are the only member of Archaea that can generate energy via photosynthesis. And unlike plants and animals that use chlorophyll for photosynthesis, they possess their own unique protein complex. Bacteriorhodopsin is the sole protein found on the purple-colored patches of the purple membrane of Halobacterium. The purple color is caused by retinol, a molecule that absorbs light at a particular wavelength and emits color as a result. In the case of Bacteriorhodopsin, the retinol is bound to an integral peptide protein that absorbs mostly green light and transmits purple light. This is why it appears purple. The discovery of Bacteriorhodopsin is kind of a big deal because up until that point, it was believed that these light-sensitive pigments only existed in animal eyes. So how does it work? When the retinol absorbs a photon of light, the shape of the molecule changes, and this change causes a conformational rearrangement in the surrounding protein. This allows for Bacteriorhodopsin to behave as a proton pump, moving hydrogen ions against their electrochemical gradient from the cytoplasm of the cell to the extracellular space. So for each proton absorbed, one hydrogen ion is taken out of the cell. The accumulation of hydrogen ions in the extracellular space activates ATP synthase as hydrogen ions move down their electrical chemical gradient back into the cytoplasm of the cell. Now, communities of hypersaline environments tend to have low complexity because competition is significantly reduced in such extreme environments where only those adapted to such harsh conditions survive. But Halobacterium is not the only organism that's adapted to survive here. The extremely halophilic red-orange bacterium Selenobacter compete for space with Halobacterium in these hypersaline environments. So does Dunaliella salina, the unicellular green algae that turns orange under abiotic stress. And most surprising, the haloviruses which actually infect Halobacterium. Now, if you remember, Archaea is not known to cause any diseases in any humans, animals, or plants, but this doesn't mean that they're immune to viruses. Halobacterium raises environmental red flags. When a railroad causeway was built in Utah's Great Salt Lake in the 1950s, it divided the lake into a north and south arm, cutting the north arm from the influx of fresh water and raising its salt content to double that of the south arm. In the north arm, Dunaliella salina and Halobacterium thrive. Despite the beautiful color displays, this was not good for the other organisms that previously existed in this ecosystem. Brine shrimp, brine flies, green algae, and all the migratory birds were no longer there. All the living communities had been reduced to the microscopic level, causing a bit of a man-made environmental disaster. When a pond in Maui turned bright pink, it serves as a warning sign that increasing temperatures and drier conditions made the water saltier, allowing for Halobacterium to thrive and for everything else to pretty much die. So the presence of Halobacterium could spell trouble for animals that rely on these important marine environments to survive. Scientists warn that our future may hold more pink lakes, that is, more Halobacterium as the planet warms.
Methanogens can be broadly defined as archaea who, unlike any other organism on Earth, produce methane gas as a byproduct of their metabolism. All methanogens are within the domain Archaea in the phylum Euryarchaeota, and from there they make up 5 orders. Ultimately, there are 150 species of methanogens. These microorganisms have earned a reputation because the methane gas that they produce is only second in line to carbon dioxide as the largest contributors to global warming. A note on methane gas. Its potency or tendency to trap heat in the atmosphere is greater than carbon dioxide. In fact, it is 25 times more potent than carbon dioxide. But the reason methane gas is second to carbon dioxide in its contribution to greenhouse gas emissions is because methane has a shorter half-life than carbon dioxide. Methanogens are usually cockeye or rod-shaped and sometimes long filaments. They tend to lack fimbriae, and they usually have flagella. Their cytoplasmic membrane contains branched hydrocarbons linked to glycerol by ether linkages. Their optimal temperature, pH, and salinity varies, which is illustrated by the fact that some methanogens are considered extremophils, living in such environments as saline lakes and hydrothermal ocean vents. While it is true of all environments, what is true of all environments that methanogens can be found in is that they are all oxygen deprived because these are obligate anaerobic organisms. The most common habitats for methanogens include wetlands, the rumen, which is part of the stomach of agricultural mammals, and landfills. Methanogens are autotrophs, so they obtain nutrition from inorganic carbon sources such as carbon dioxide. Their process of anaerobic metabolism is called methanogenesis because, as the name predicts, methane is produced as a byproduct of their metabolism. Other substrates besides carbon dioxide that are used by methanogens include hydrogen, methylated compounds, and acetate. Methanogens live in symbiosis with certain microorganisms while living in competition with others. One symbiont is the protozoa, who release hydrogen gas as a byproduct of their metabolism. Methanogens then use that hydrogen as a substrate to their own metabolism. Protozoa, in turn, benefit from methanogens' utilization of hydrogen gas because when accumulated in high amounts, the hydrogen becomes toxic to protozoa. A common competitor to the methanogen is sulfate-reducing bacteria. These act as competitors because they also use hydrogen as a substrate, but they tend to outcompete methanogens because they have the ability to utilize a variety of other substrates. As mentioned before, methanogens are found in the rumens of cows and other agricultural mammals. It is here that species Methanobrevibacter ruminantium predominantly reside. The methane gas that these archaea produce gets either belched or flatulated by the cows, ultimately leading to its release into the atmosphere. Due to the mass agriculture of cows, where there is estimated to be 1.5 billion cows living on Earth, this has resulted in the release of 300 billion liters of methane gas every day. An estimated 60% of methane emissions today are the consequence of human activities, i.e., from landfills and/or agriculture of cows, while the other 40% of methane emissions comes from natural sources such as wetlands. In conclusion, the methane gas produced by methanogenic Archaea has environmental implications, and it is important that humans acknowledge their role. Providing viable homes for methanogens, such as cows, directly contributes to the production of methane gas. Therefore, it is crucial that humans utilize and practice sustainable agriculture whenever possible.